Welcome to this comprehensive video on Therapeutic Drug Monitoring, or TDM, which is vital for optimizing patient outcomes. This guide is tailored for B.Pharm, M.Pharm, and Pharm.D students. Our learning objectives are to define TDM, apply pharmacokinetic principles, identify narrow therapeutic index drugs, and solve exam-level questions on monitoring and dose adjustments. TDM is the clinical practice of measuring specific drug levels in the bloodstream. The goal is to maintain a constant concentration within the therapeutic range, the sweet spot between efficacy and safety. The therapeutic range is the core principle. Narrow therapeutic index, or NTI drugs, have a small margin between effective and toxic doses. Examples include digoxin, warfarin, and lithium. TDM is necessary due to PK variability. Factors like patient demographics, organ function, genetics, and drug interactions all affect absorption, distribution, metabolism, and excretion. The correct timing for sampling is crucial. We measure trough levels, the lowest concentration, to check for toxicity, and peak levels for efficacy, typically after reaching a steady state. Let's review common NTI drugs. Digoxin is for cardiovascular issues. Phenytoin and carbamazepine treat seizures, and lithium is used for bipolar disorder. Please memorize the therapeutic ranges. More NTI drugs include warfarin, where we target an INR of 2 to 3, and antibiotics like gentamicin and vancomycin, which require monitoring for severe infections. Theophylline is used for respiratory conditions. Here is a pro tip. Always check the patient's creatinine clearance before interpreting TDM results for renally excreted drugs, such as vancomycin. Take a moment to review this key concept.
The TDM process involves 3 main steps. First, patient assessment. Second, sample collection with critical timing. And third, laboratory analysis, often using HPLC.
TDM is a continuous feedback loop. We assess, measure the drug levels, compare the result to the therapeutic range, and adjust the dose as needed. The case scenario shows an adjustment due to toxicity. Challenges in TDM include analytical limitations like the cost of HPLC and delays in turnaround time. Patient factors, such as non-compliance and variable pharmacokinetics, also pose challenges. In the exam corner, we focus on GPAT-relevant questions. Warfarin requires TDM. The therapeutic range of digoxin is 0.8 to 2.0 nanograms per milliliter, and HPLC is the gold standard for analysis. Use the mnemonic, "Warning!" These drugs pose lethal consequences to memorize NTI drugs: warfarin, theophylline, digoxin, phenytoin, lithium, and cyclosporin. Recommended texts are also listed here. To summarize, TDM balances efficacy and safety for NTI drugs. Success relies on accurate PK principles and sample timing. Remember to treat the patient, not just the number, and memorize the key therapeutic ranges.